All right, so we're going to talk a little bit about molecular biology, just an introduction. So molecular biology focuses around DNA and the application of DNA. So it tracks the path from protein expression, right, to protein. So things that we're interested in, cell signals, right? So remember, cell signals are the way that things enter the cells. So how we can activate cells, how we can activate DNA. Replication, right? So DNA polymerases are of interest. Transcription, remember how I read DNA. Translation, how I make DNA into proteins. Then how we modify those proteins to make them functional. So this whole path from DNA to protein is kind of the focus of what we want to do here. Right, so it all starts with DNA, like DNA and RNA. Let me make this a little bit. Right, so DNA, just to review for you, DNA is double-stranded, so both strands are anti-parallel, right? So DNA is double-stranded. Right? So we say that we have a 3' end. So one goes 3 to 5, the other goes 5 to 3. So they're anti-parallel, right? So they're running in opposite directions. They're not exactly identical, but they're complementary. So these are anti-parallel, and we can say that and complementary. Right, so they're anti-parallel and complementary. What we say complementary is A is always bound to G, and C is always bound to T. All right, so let's talk a little bit about DNA versus RNA. So we talk about the primes. So if you look at these pictures right over here, let me make this a little bigger. All right, so if you make these pictures a little bigger. So the carbons are named starting from the right-hand side of the oxygen. So if you see that we've got the 1', the 2', the 3, the 4, and the 5. Same thing with the RNA. 1', 2', 3', 4', 5'. So components of DNA, right? So we have deoxyribose, right? And it's deoxy at the 2' carbon. So it's deoxy at this 2' carbon right here, right? Because you'll see that I have an H here. And if you look at RNA, RNA has ribose, right? Because at the 2' carbon, I have a hydroxyl, right? So at the ribose, I have a hydroxyl. Right, that's it right here. So OH is a hydroxyl. Right, so, and the reason why that's important is I can add things by dehydration synthesis to any OH, right? Two OHs that come together, I can take water out and make them bond. So DNA is set up so it can't be modified at that 2' carbon. If you look at the 1' carbon, it can't be modified there either. Right? So, and at the 4', I can't be modified there either. So if you look at DNA, DNA can only be changed at the 3' and the 5' end. Right? The 5' end because you've got a phosphate group there. Right? RNA can be modified at 3 different sites. Right? And that's why RNA it's edited into so many different useful tools for DNA because it can be modified more. Right? So things to know about DNA. Right. So things you might want to know about DNA. So, like I said, it's anti-parallel. Right. It's complementary. Let me just make another structure. It has a sugar nitrogenous bases on the inside. All right, so if we look at this picture here, my sugar phosphate backbone's on the outside. So I've got a phosphate and a sugar. I've got a phosphate and a sugar. Right? So all those are on the outside. And this one, 3 to 5 here, and it's anti-parallel there. My nitrogenous bases are my A, T, C, and G, and they're on the inside. Right? And you can see that C is paired to G, and T is paired to A. C and G have 3 hydrogen bonds that join them, and T and A have 2 hydrogen bonds. Think about it. C and G both bind with 3, and T is the first letter of 2. So if it binds to T, it has 2 bonds. If it binds to C or G, it has 3 bonds. We have purines and pyrimidines, right? So A and G are purines. Right? And these purines, if you look at A and G here, have 2 rings. T and C are pyrimidines. And they have 1 ring. So they're classified as purines and pyrimidines. Right? You can tell them by the rings, or you can remember that A and G, right? And we told in college that agriculture is a pure science, and agriculture is AG. So it was kind of stupid, but I still remember that to this day. All right, so DNA replication. So if we talk about DNA replication, DNA replication. So DNA, here's the simple rule, is read 3' to 5' and assembled 5' to 3'. Okay? So DNA is read 3 to 5 and assembled 5 to 3. All right. So if we look at our picture here, there's some enzymes that you need to know. All right. So we have RNA primase, and this adds for RNA primer. We have DNA polymerase. And this adds right, as nucleosides 3 to prime. We have DNA ligase, and this ties DNA strands together. So there's 3 enzymes we're going to talk about today. And there's other enzymes here. There's an enzyme that goes around your DNA here. And this would be topoisomerase. Right? And this relieves supercoiling. That's another important one there, right? Because DNA is a alpha helix, and if I just tried to pull it apart, it would make knots on both sides. Right? So that's an important one, and that topoisomerase moves in the direction of the replication fork. So, for DNA synthesis, right? So if I have 2 strands of DNA, right, I'll have sites called the origin of replication. So the origin of replication, and that's where DNA is that's where replication starts. So I've got 3' to 5' and 5. Oops. I would do that. And 3' to 5'. So this thing right here is called a replication bubble. And then half of it here is a replication fork. Right? So we would get, so it's read 3 to 5, so I would get an RNA primer. And I'd have another RNA primer right there, right? And now my replication is going to go in this direction, right? It's always read 3 to 5, and I'm going to apply it 5 to 3, right? So it's always heading toward a replication fork, right? Now, I will add another RNA primer. Right here. Oops. So another RNA primer will be there, and another RNA primer will be there. And these ones will be heading this way. So you see the problem is, oops, and I forgot there's one more enzyme that's super important. There's another enzyme here. And this one is called helicase. Helicase opens DNA. And I'll have a helicase there. And those helicases are moving in that direction, so they're widening the replication bubble as it opens. Right? Right, so I have my DNA, or my RNA primer here, and so on RNA primer, I'll get DNA polymerase 3. Right? And it will start replicating DNA. Right? So it'll start moving in the direction of that arrow, and it'll be making DNA. Right? Another one here, I'll have DNA polymerase 3. Right? And it'll start making DNA in the direction of that arrow. I'll have another one here. And I'll have another one here. So there's a lot of DNA polymerase, and it's all working at the same time. Now that DNA polymerase, if we're on the short side, right, it'll make it until it hits that RNA primer, right, and then it'll stop. Right, so this right here, I'm just trying to make it a different color. This right here is what they call an Okazaki fragment. So that short fragment that's made, this would be another one over here. The short fragment that's made between the fork and the ORI is called an Okazaki fragment. Right? So the Okazaki fragment will be made, and when that DNA polymerase hits the primer, it'll fall off. Right? So the Okazaki fragment DNA polymerase 1 will replace RNA primer, and then DNA ligase. DNA ligase will turn the kinase together. So, like I said before, we're really interested in enzymes because enzymes do a lot, right? So that's the whole DNA replication structure, and that's what we talk about here, right? So I do that here, and I give you this right here. So remember, so this picture right here that I'm showing you, right around here, that's the replication fork. Let me erase my topoisomerase. Okay, so that's a replication fork here. And that replication fork, I lost my pen. So that replication fork here, now you'll see that the 3 to 5, so I've got that red line there is my growing DNA strand. I've got my DNA polymerase 3. And it'll keep going in that direction on my leading strand as it's opened up, right? So remember we have a little enzyme here, helicase, and that helicase is opening up that replication fork, right? As it opens up. Now, as it opens up, it creates more and more of these little Okazaki fragments, right? And the Okazaki fragment is just that lagging end where it's getting rid of those little short pieces. Right. Okay. Oops, I don't want to move that. Right. So the assumption for molecular biology is that all DNA is the same. So every organism right? We all have A, C, T, and G, right? So we all have that, right? RNA as U of T, right? So all organisms, whether you're a bacteria or you're a human, we all have the same basis. And if we all have the same basis, then I can genetically engineer, right, and insert DNA from different species into each other because our DNA is all the same. We all have origins for replication. We all have promoter sites, right, where we're going to read DNA to make proteins. DNA packaging is similar in eukaryotes, a little dissimilar in prokaryotes, but it doesn't matter. So DNA packaging, just know that DNA packaging, so this allows for differential gene expression. Right, so remember, every cell in your body has the same DNA, right? But each cell is not expressing DNA the same. It's because they're all packaged differently, right? And it exposes areas where we want genes to be transcribed, and it hides areas where we don't want genes to be transcribed. So important things here, right? So chromatin, remember chromatin is loose DNA, is packaged around proteins called histones, right? And those histones, right, if you look at this picture down here, form complexes of 8 proteins, and the DNA is wrapped around them. And if the DNA is wrapped around histones, it can't be read. It's hidden. And to tell you how small DNA is, the 8 proteins there with DNA around is only 10 nanometers wide, right? So, pretty small. But what we get is, we get euchromatin, and heterochromatin. From this packaging. So euchromatin is loosely packaged. And then heterochromatin will be tightly packaged. So I would be reading genes in euchromatin areas, and I wouldn't be reading genes in heterochromatin areas. So if you look, we have DNA, right? We have nucleosomes. So this would be a picture right here of a nucleosome, right? This would be a nucleosome, right? Because I have histones and DNA, right? Chromatin, right, would be just the loose package. And I make these loops, and the end of these loops would be my euchromatin, right? And then this chromosome here, right, this picture of a chromosome is we only see in mitosis. So essentially mitosis becomes condensed, and it's all heterochromatin at that point, right? So active regions are only transcribed when needed. All right. That's a very brief and very general description of DNA. All right. Talk to you soon.